here. So this blue one is the respiratory diverticulum. So first of all, how the gut knows that this is the territory where this diverticulum has to appear. Well, there is something, a substance called retinoic acid, which is expressed at this region. And it induces the expression of a transcription factor called TBX4. And this triggers the appearance of this respiratory diverticulum. And these kind of evaginations are called tracheoesophageal ridges, which are here. And then this is just a continuation anteriorly from the foregut at the beginning. But later a septum appears, which will separate this future esophagus, which is derived from the foregut also, and the primordium of the respiratory tract. And then this respiratory diverticulum branches. These two branches are called lung buds, and these will elongate and branch further. So this is how the respiratory system develops. And this is a kind of timeline. And 4 periods can be distinguished in the development of the lungs. The first period is from week 5 to week 16, and this is called pseudoglandular period because histologically the developing respiratory system looks like a gland, a primitive developing gland, that's why the name. And during this period, the respiratory tract develops until the terminal bronchioles, okay? And then the canalicular period comes, referring to the fact that a lumen appears in these glandular epithelium, glandular-like epithelium. This is between week 16 and 26, and during this period, the respiratory tract develops further until the future alveolar ducts. And then the next period is terminal sac period from week 16 to birth, and as the name suggests, this is where terminal sacs, if you like, primitive alveoli appear, but not matured alveoli really. The final period is the alveolar period when real matured alveoli start to appear from months 8 to childhood. This childhood means something like 10 years. So it also means that about only 16% of the total number of alveoli appear by the time of birth. So the vast majority appears later during early childhood up to year 10 approximately. So that's the alveolar period, and of course here during alveolar period here you can see the further branches. So lobar and segmental bronchi appear, 3 lobes on the right side, 2 lobes on the left side, and this red line is the pleura, the visceral pleura, which becomes continuous with the parietal layer of the pleura, of course. Here you can see the visceral and parietal layers of the pleura, and you can see here that these two are really continuous with each other. Now, obviously during the development of the lung, one of the most important points is the development of type 1 and 2 pneumocytes. Type 1 cells appear first, and you know these are involved in gas exchange. Type 2 alveolar cells start to appear from around month 7, and they will start to produce surfactant. And before term, the surfactant which is produced is not enough. So that's why if a baby is born prematurely, especially if it is born before the 7th month, before the end of the 7th month, the major problem usually breathing for the infant, and this problem comes from the lack or not enough production of the surfactant. And this problem is called respiratory distress syndrome, or RDS disease. The old name is hyaline membrane disease, but the modern name is respiratory distress syndrome. And as I said, the problem is the lack of surfactant, and therefore when the baby is doing expiration, alveoli tend to collapse, and that's the problem, because normally it is prevented by the surfactant, which decreases surface tension. Now, nowadays most babies survive because steroids and artificial surfactant can be given to the newborn to overcome this problem. And one more interesting aspect here, that because breathing is a major thing when the baby is born, a signal is coming from essentially lungs to the wall of the uterus, because the baby, when it is developing, the fetus, it is within the amniotic fluid, and this amniotic fluid goes into the developing lungs as well, and therefore the surfactant appears in the amniotic fluid. And when this surfactant concentration goes up really, which happens just 2 weeks before the baby is born normally, this increased amount of surfactant will trigger macrophages, which also present in the amniotic fluid, and they will migrate through the chorionic plate and enter the wall of the uterus. And there they will trigger production of prostaglandin, which has an effect on the smooth muscle of the wall of the uterus, forces contraction. So this is a signal. If enough surfactant is produced, these contractions starts, and this is the painful contractions which eventually will lead to delivery of the baby. Well, this is not the only thing which contributes to this powerful muscle contraction. Another powerful substance is the oxytocin, a hormone, what we will learn later. Okay, so yeah.
Isn't the oxytocin what's released in pain?
No, I don't. All these are variations how abnormal separation between the respiratory tract and the future esophagus may happen. This is the most frequent. And these problems can be divided into two major categories. There are athresias and fistulas. Atresia is something which should develop but doesn't appear at all. So for example, there is a kind of atresia here because this continuation between the proximal and distal part of the esophagus is missing. And fistula is when an abnormal connection is present between two structures which shouldn't be there. Like here, the distal part of the future esophagus remains connected to the trachea. And there are many other variations, as you can see. So these are kind of major problems in addition to the problems with the lungs. Yeah.
What exactly the cause of the fistula, the connection between?
Because if you remember here, this is the start of the development. So a septum needs to appear between the future esophagus and the respiratory tract. So that septum is abnormally formed or didn't form at all. Okay. So that's a kind of summary, much easier than the development of the heart. Yes?
There is some specific cells that, like progenitors, that create the trachea, or
I don't know about progenitor cells. Well, much more is known about the genetic background actually. I don't want to go into such details, but you can find some details in your book. Here are some details. What I mentioned is the retinoic acid which appears at the site of the future respiratory diverticulum, which will induce, upregulates TBX4 production, which is a transcription factor.
and this is the process that we have to know. Or
Well, in principle, what you need to know is everything what is in your official textbook, what is discussed here, and what is discussed in the lecture hall. Whenever you ask this kind of question, I will always answer this, because if I say anything else, I'm lying. Obviously, we don't start asking you about the dozens of genes which are involved. Okay. So I try to give focus for those parts, which is really the focus during exams as well. But remember what I just said, what is in the official curriculum among the requirements. Okay. Now, so then we need to start the alimentary canal. And let's start in the proximal part, esophagus 45. It's a cross-section, hematoxyanosin staining. And you have seen this in the previous semester when we learned the types of lining epithelia. So hopefully you remember what kind of lining epithelium is here. Yes, certified squamous non-keratinizing epithelium. So the lumen is quite irregular, and the total thickness of the wall is quite substantial. So let's look at the epithelium. From here down to here, this is the epithelium. Certified squamous non-keratinizing epithelium. You can see squamous cells on the surface. And you can see that there are kind of detachment here. Cells tend to detach from the surface. Well, this is partly a natural process because when these surface cells die, they desquamate from here. So they get detached from here. But why we can see these detached cells, another reason is also possible, which is an artifact. During sectioning, we just artificially removed these surface cells. We cannot tell which is the case here, both are possible. And we learned that this epithelium can be divided into 3 sublayers. The most basal sublayer, a single cuboidal cell row, is called stratum basale or germinativum. This is where new cells are born. That's why the name germinativum. Basale refers to the location. This is the most basally located cell row. And from here upwards we have quite a lot of cell rows. As long as we reach those cell rows which have really squamous nuclei, I would say here, those will give the most superficial sublayer, but up to this, from here, the middle sublayer is called stratum polygonale or stratum spinosum, because we have polygonal shaped cells here, and spinosum is referring to the spines. Spina is spine in Latin, that's why the name. And this name refers to the fact that these cells in a better specimen, in a better microscopes, in the stratum spinosum look like spiny. We cannot see them here, but in reality they are not spiny in vivo. Why they look like spiny in a better specimen, in a better microscope? It has 2 reasons. One is that these cells are usually interconnected by desmosomes, very mechanically stable intercellular junctional complexes which can hold them together despite the forces which arise during dehydration. So this is one reason why the cells in the microscope may appear spiny, but the other reason is the dehydration itself. During dehydration, cell borders tend to get shifted farther apart, except where desmosomes hold them together. Okay? And these desmosomes are in spot-like surfaces, so not homogeneously distributed over the surface. Desmosomes are present in certain spots, elsewhere they are not present. So when we have these spots full of desmosomes, though, at those territories, cell membranes cannot get shifted despite the dehydration, because desmosomes can hold them together, and these are the territories where these artificially appearing spine-like processes become visible. So that's the origin of the name. And the most superficial few cell rows where we have really flattened cells, like here or here, those cell rows collectively refer to the stratum planocellulare. Plano means plain, flat in Latin, that's why the name. So referring to the flattened nature of cells in these cell rows. Okay, another general thing that this epithelium, especially the most apical part, looks very pale, paler than other types of lining epithelia. This is due to the fact that these cells contain lots of glycogen, more glycogen than the cells containing other types of lining epithelia. So certified squamous non-keratinizing epithelium contains a higher concentration of glycogen than other types of epithelia, and this is true not only here in the esophagus but also in other organs where we have this kind of epithelium. For example, in the vagina we also have certified squamous non-keratinizing epithelium, and it also looks like pale. So remember for this when we will study.
Can you call the outer layer also superficiale?
Yes, that's another name, stratum superficiale. It's also in use. Yeah. Okay. Now, this is a rather resistant type of epithelium. There is just one even more resistant type in the human body, the stratified squamous keratinizing version, which covers our skin. So this is the second most resistant epithelium. And this resistance is needed because different stresses act on this epithelium. For example, mechanical stress, when we swallow a food ball, which is, let's say, we eat toast. It's a rather rough substance even after chewing and even after mixing it with saliva. It can still cause a considerable friction here over the surface when we swallow it. So this is a mechanical stress for this epithelium, and this epithelium needs to be able to stand this stress. To be able to stand this stress, desmosomes hold cells together, as we discussed. And there is another stabilizing structure here, these so-called connective tissue papillae underneath. So piece of the connective tissue layer evaginate towards the epithelium, forming these peninsula like this, which are called connective tissue papillae, making the interface between epithelium and connective tissue in between the basement membrane. So this connection is wavier, which increases the total surface area where these different layers can attach. And if these layers can attach over a higher surface area, then firmer attachment can be achieved. So one function of these connective tissue papillae is to stabilize tissues, not to allow the epithelium to shift relative to the underlying connective tissue layer. But mechanical stress is not the only stress which occurs here, because sometimes we drink something which is acidic, like grapefruit juice. It has a relatively low pH value, so that's a chemical stress. Or we drink alcohol, that's also a chemical stress. But this epithelium suffers from thermal stress as well, because sometimes we drink something very hot or very cold. That's a thermal stress. So different stresses are here. So back to this connective tissue papilla. Apart from the mechanical stabilization role, it has another role. Can you remember? This is repetition. Do you mean that blood is flowing from here to the epithelium?
No, I think it's
By
more
diffusion.
Diffusion. So nutrients and oxygen can diffuse from blood vessels, which are in the connective tissue papilla, towards the epithelium to nourish and oxygenate the epithelium. This is needed because the epithelium is avascular, there is no blood vessel inside. And when we have connective tissue papilla, these small capillaries can get closer to the superficial layer, so the route for diffusion gets shorter, which means the nutrient supply and oxygen supply will become more efficient.
Also immune response, no? Also immune response.
Immune response? What do you mean? I don't understand.
There is white blood cells in the connective tissue papilla.
Yes, but it happens. For example, here maybe you can see this nucleus. This is a heterochromatic round nucleus, which is seemingly different from the nuclei of epithelial cells. Okay, it's more heterochromatic and round and smaller. So that's probably a lymphocyte nucleus. So sometimes lymphocyte may migrate into the epithelium as a part of immune response. And obviously it is also easier for them if the source, the blood vessel, is closer to the epithelium. But yeah, you are right, but I wouldn't say this is more important than the previously mentioned two. because this is just rarely occurring, at least in the esophagus. But as you can see, it does happen sometimes. Okay, and in relation to this, I should mention structure like this in the epithelium, which is an eosinophilic island-like structure, giving the impression that there is a connective tissue island within this lining epithelium, which is absolutely false impression. This is also a part of a connective tissue papilla, and it is continuous with this connective tissue layer. The only thing, we cannot see the continuation because it is outside of the plane of sectioning, which is very thin. Okay, and here also where you can see this reddish tint, those are signs of connective tissue papillae. You know, they are a little bit curvy and they are not parallel with the plane of sectioning, that's all. Okay, so if we leave this epithelium, there is a basement membrane which is very thin, so it is not really possible to distinguish it, but it should be here somewhere. And on the other side of this basement membrane, we have this connective tissue layer here, which is very eosinophilic because of the presence of many collagen fibers. So this layer belongs to the mucosa, just like this epithelium belongs to the mucosa, this term what we introduced last week. So if we have an organ with a lumen, then the lumen is lined by a mucosa or mucosal membrane, which has 2 or 3 sublayers depending on the organ. In the trachea, we had 2 sublayers only in the mucosa, epithelium mucosae and lamina propria mucosae. So we can say it is an incomplete mucosa in the trachea. But here we have a complete mucosa because we have all 3 sublayers. So we have the epithelium mucosae like this. We have the lamina propria mucosae, this. And then the third sublayer is this muscularis mucosae, which is a smooth muscle layer. Okay, so we have a complete mucosa here.
This whole section between the epithelium and the muscle, this whole is the lamina propria, all of this?
No, lamina propria is this.
Yeah, this whole thing is
between the epithelium and the muscularis mucosae.
It's pretty thick, right? It's like compared
Lamina
to
propria.
Yeah.
Well, the muscularis mucosae is much thicker, and also the epithelium is thicker. So actually, this is the thinnest layer in the mucosa, at least here. But it is variable from organ to organ. Okay. And please note that the lamina propria is more reddish than the smooth muscle, because this is true generally, which helps to distinguish the muscle from the connective tissue, usually in a hematoxin eosin stained section. Now, this muscularis mucosae contains smooth muscle cells, mostly in oblique sections. The reason is that actually most of the smooth muscle bundles here have a spiral course along the longer axis of the esophagus. And that's why if we look at them in cross-section, most of them appear in kind of oblique sections. Well, sometimes it is almost cross-section, like here. And this is almost longitudinal section, so it is variable, but the major tendency is that this is spiraling. Okay, now if we leave this muscularis mucosae,
usually the connective tissue papilla is reticular fibers, and the submucosa is.
no, you cannot say the connective tissue papilla is reticular fiber. There may be reticular fibers there, but there are many more things there.
The blood vessels and everything.
Blood vessels, nerves, connective tissue cells. So it is far more than just reticular fibers.
For sure, but if we're talking about the fibers, it's supposed to be reticular fibers at the submucosa?
No, not exclusively reticular fibers, no. No. There are other types of fibers. Mostly collagen, but not only reticular fiber. Okay, so if we leave this muscularis mucosae, we enter another connective tissue layer here, which is already not part of the mucosa, but it is underneath the mucosa. That's why we call it as submucosa, sub, underneath, okay? Submucosa. Well, this looks like more or less empty territory, but this appearance is mostly an artifact due to separation of the connective tissue layer from this outer muscle layer. So submucosa contains glands like this. Mucus and serous secretory units, mostly mucus. Here these are 3 mucus secretory units, but serous secretory units may also occur. So we can say there are small salivary glands in the submucosa. And obviously ducts lead from these glands to the surface because these are exocrine glands. Well, these ducts sometimes visible. For example, here look at this structure. This is a very big duct. Also this one. Here you can see the secretory units. Also here, and this is also a duct here. So look at the lining epithelium of this duct. This is definitely a stratified epithelium, which is characteristic for the bigger ducts, as we learned. Okay. Here you can see a duct which is almost longitudinally sectioned and shows that it really goes towards the epithelium, okay? And actually it will pierce the epithelium. Okay.
Sorry? It starts from the submucosa?
It starts from the secretory units, and the secretory units are mostly in the submucosa. Some may be in the lamina propria as well. But most of them are in the submucosa.
Thank you.
Okay. Now, so if we go back to the submucosa, which is here, there is a nerve plexus here in the submucosa, which is rather underdeveloped here at the level of the esophagus. You can see a very tiny piece of this nerve plexus, which is called plexus submucosus or submucosal plexus, which consists, plexus means network, and it consists of axons and ganglion cells, which are special nerve cells. Well, ganglion cells, probably I won't be able to show you ganglion cell body here, but I will be able to show it elsewhere. Okay, so this plexus, this submucosal plexus is also called Meissner's plexus. It's an author's name, capital M and double S inside, Meissner's plexus. And postganglionic fibers, which arise here from the ganglion cells, innervate the smooth muscle cells of the muscularis mucosae and may also innervate glands. So this plexus is an autonomic plexus, mostly parasympathetic, arising from the vagal nerve. Okay. Now, if we leave this submucosa, we enter a muscle layer which may be called as external muscle layer, very thick. And in this section, this is entirely smooth muscle as well. And it can be divided into two sublayers. An inner sublayer, which is the inner circular sublayer, because smooth muscle cells here circularly surround the lumen. And because this is a cross section, that's why here smooth muscle cells are seen in longitudinal view. Okay. So inner circular sublayer, this one, and outer longitudinal sublayer, where smooth muscle cells are oriented parallel with the longer axis of the esophagus. And that's why we see them mostly in cross-section in this sublayer. So if I enlarge, you will see here these round-looking nuclei, which are the cross-sections of smooth muscle cell nuclei, which are rod-shaped, but in cross-section they look like circular. Okay. And between, yes?
Can you say the names of the layers?
Inner circular, outer longitudinal sublayer.
Okay.
And between these two sublayers, there is another nerve plexus which is a bit more developed than the submucosal plexus. You can see a piece here. This is called myenteric plexus. Myenteric plexus. Again, composed of axons and ganglion cells. And here you can see nice ganglion cell body, like this. Basophilically stained cytoplasm. Euchromatic nucleus with the nucleolus inside. This dark spot inside is the nucleolus. And the basophilia in the cytoplasm is because of the high rate of protein synthesis which occurs here. This is generally true for all nerve cells, so lots of RNA in the cytoplasm. Euchromatic nucleus refers to the high rate of gene expression, which is also characteristic for nerve cells in general. And here you can see another example. This is also a ganglion cell with the euchromatic nucleus, and inside you can see maybe 1-2 nucleoli. Okay. Here, this plexus mostly innervates this external muscle layer, smooth muscle cells inside. Okay. And the outermost sublayer is a connective tissue layer again.
For the myenteric plexus, is it parasympathetic or
Parasympathetic. And it also has an Ausos name called Auerbach plexus. Auerbach, capital A at the beginning. Here this is also belongs to the myenteric plexus. This is just the peripheral nerve, as you can see. Also here. Here no ganglion cell is identifiable, just nerves.
Can you see the vagus nerves?
These are branches of the vagus nerve, because that's the parasympathetic innervation here. Yeah. So the outermost tissue layer is adventitia, the loose connective tissue. Adventitia. Okay. And what else? Okay, one more thing. Here, the muscularis externa or external muscle layer is entirely smooth muscle here in this section. And from this fact, we can conclude that this sample is from the lower part of the esophagus. The reason is that in the upper part, near to the pharynx, this external muscle layer is composed of striated muscle, visceral striated muscle, and no smooth muscle there. Then if we go to more distal levels, then striated muscle and smooth muscle both are present. And then finally, striated muscle disappears entirely and only smooth muscle remains. This is the lower part, and this is what we have here. And it must be here, it must be like this, because the esophagus is connected to the stomach, and the stomach wall contains entirely smooth muscle in the muscularis externa, and obviously this continues with the muscularis externa of this esophagus. So if the stomach has only smooth muscle, then the lower part of the esophagus has to have only smooth muscle as well. Here you can see peripheral nerve sectional profiles here again. Okay.
Professor,
Yes.
please show again ganglionic cell.
What?
Ganglionic body.
Ganglion cell body.
Yes.
Yes. In the myenteric plexus,
Yeah.
this is where I found them. So this one, or this one, or this one.
The most pathogenic will be the ganglion, right?
This is the cell body, the cytoplasm. This is the nucleus.
Okay.
And inside the nucleus we have, I don't know, 2, 3 nucleoli, the dark spots.
So all the shape is the ganglion, right?
These are ganglion cells,
Yeah.
okay, which don't necessarily form a full ganglion,
Okay.
but may form.
Thank you.
If it's the lower part of the esophagus, there's not supposed to be also a serosa there?
Good question, because the stomach is covered by peritoneum, so we call it intraperitoneal organ. So this is the lower part, but not the lowermost part. So it is not the part which is directly connected to the stomach, a little bit above. And once you mentioned, this is a good point to emphasize that in the wall structure of organs with a lumen, the outermost tissue layer is either adventitia or subserosa and serosa, depending on whether or not the organ is covered by a serous membrane. Serous membranes are the peritoneum, the pleura, and the pericardium. So if an organ is covered by one of these serous membranes, then the outermost tissue layer of the wall is called subserosa and serosa. We will discuss it just in the next section in relation to the stomach. So let's take the stomach.
So whenever you swallow, you can control is the striated muscle, and whenever you just can't control anymore, the esophagus is where only the smooth muscle goes?
Well, smooth muscle is definitely involuntarily controlled. The striated muscle can be controlled voluntarily in some cases, sometimes involuntarily. So, you can decide whether or not you want to swallow something, but also it happens involuntarily when you are preparing the food you are chewing. It also comes as a reflex. So both.
So the striated.
Because you can swallow voluntarily and involuntarily as well. Yeah. So let's take the stomach section. We have 2 hematoxene stained version, the 47 under line 1, 47 under line 2. I will open both. And these are the same, just one of them is paler than the other one. Now, the stomach macroscopically can be divided into 4 parts. Can you name those parts?
Cardia,
Fundus, cardia,
Body,
body,
fundus.
or
Yes, so cardia, body, fundus, and pylorus. Now, these sections are either from the corpus or from the funding part. We cannot tell from which part because histologically these two parts are different. But these sections cannot be from the other two parts because the glands are different there. We will return to this point. Now, so this is the paler one, 47 underline 2. And if you look at the surface, some piece of the stomach content remained here and it is stained. This is due to the fact that here we have lots of mucin in the stomach, and probably here non-formalin fixative was used, and that's why mucin was at least partly preserved and would be stained here on the surface. If you look at the real tissue surface of the stomach, you can see these kind of evaginations, like these.
But there are cells on the mucin. Like if you show where the food is, or like the bolus, in the top right, yeah, there's cells there.
This is just dust.
No.
Here. Yeah, you know, during sectioning, again, some cells may be artificially shifted from the surface. That's not really important. So these evaginations of the surface are called areola. Areola. The plural is areolae, A-E at the end, because this is Latin, so this is the plural. Areolae. And between areolae, we have depressions like this, or like this, or like this, or like this. These are the gastric pits, And the lumen of the gastric pit is obviously continuous with the lumen of the stomach. Okay. Now, if we look at the surface epithelium, the lining epithelium, it is appearing as simple columnar epithelium, and all columnar cells here produce mucin. So this reddish-like substance here, now the mucin. This mucin is different from the mucin which is produced by goblet cells, that's why the different color. And by the way, there is no goblet cell in the stomach at all, no goblet cell in the stomach. So because all these columnar cells produce mucin in the lining epithelium of the stomach, we call this epithelium as simple columnar mucin producing epithelium. Okay? To refer to the fact that all these columnar cells produce mucin. Now, underneath, obviously, we have a basement membrane. And underneath the basement membrane, we have a connective tissue layer. So that's the lamina propria. And this lamina propria in the stomach contains lots of lymphoid cells, cells which belong to the immune system, like these. For example, this is a lymphocyte, a heterochromatic round nucleus, almost no cytoplasm around. Okay, this cell looks like a cell which has an irregular cell boundary, eosinophilic cytoplasm, and a roundish, slightly oval euchromatic nucleus. What cell can it be? Macrophage, histiocyte. Lots of enzymes in the vesicles of the cytoplasm, that's why the eosinophilic staining. enzymes are proteins, which break down the phagocytose substances. Okay, if you look at this, maybe,
Also macrophage.
probably not, because here the nucleus is round and extrinsically located and the cell is oval shaped, and maybe this is a slightly basophilic cytoplasm, so that's why it is a plasma cell probably, producing antibodies. But there are other cells here, white blood cells, all kinds of white blood cells, granulocytes, monocytes, lymphocytes may appear, also plasma cells and macrophages. These cells all belong to the immune system, lymphoid cells if you like. In addition to these lots of lymphoid cells in this lamina propria, there are reticular fibers, these thin fibers which can branch. This is the most characteristic fiber type here. So we have two characteristic features of this connective tissue layer, the lamina propria in the stomach. Presence of lots of lymphoid cells and presence of lots of reticular fibers. And that's why the type of the connective tissue is called as lymphoreticular connective tissue. Lymphoreticular connective tissue.
It's that same way until the colon, right? From small
First of all, the lamina propria of the esophagus is still not lymphoreticular. So lymphoreticular lamina propria starts here in the stomach. And it continues into the small intestines and large intestines, and it will change again in the anal canal, what we will see later. So most part of the alimentary canal, the lamina propria, is given by lymphoreticular connective tissue. Okay? Well, not all part. Here you can see these reticular fibers. Alright. Now, in the lamina propria we have glands, tubular glands, which span the entire thickness of the lamina propria. And then comes the muscularis mucosae here, smooth muscle. So we have a full mucosal membrane here in the stomach as well, with all three sublayers, just like in the esophagus. Now let's go back to the glands.
There is an option that it's serous glands.
Hmm?
There is serous glands here.
Well, it is not that easy. We don't use this terminology here, mucous or serous, because here in these tubular glands we have multiple cell types. And on top, these cells, different cell types, are not homogeneously distributed along the length of the gland, but inhomogeneously distributed. Another thing, that several of these tubular glands open into the same gastric plate. Here, this is one opening, another opening, another opening, another opening, here also. So as you can see, 7, maybe 10 tubular glands, gastric glands, may open into the same gastric plate. And at the very top, this gastric pit is lined by the same type of simple columnar mucus producing epithelium what we could see on the top surface. It goes into the gastric pit as well. But when these glands open into the gastric pit, here these glands, in this upper region of these glands, is given by these pale stained cells, which are mucus producing cells. These are called mucus neck cells, because in the neck they are predominantly in the neck region in these glands. Mucous neck cells. But if you compare the cytoplasm of these mucous neck cells with the cytoplasm of these surface mucus producing cells, they have different color, reflecting the fact that they produce different kinds of mucins. Okay? Different kinds of mucins. So these mucous neck cells are mostly in the upper part in the gland. Now if we go deeper in the gland, these highly eosinophilic cells appear like this, or like this, or like this. These are the parietal cells with very eosinophilic cytoplasm. Now these cells produce hydrochloric acid, well, actually not in the form of hydrochloric acid, but hydrogen ion and chloride ion, both pumped out from the cell by using active processes, energy-dependent processes, by using pumps, special proteins, carrier proteins embedded into the cytoplasmic membrane. And because these has to be driven by energy, lots of mitochondria are needed in the cell to produce that much energy, and mitochondria cause primarily the eosinophilia in the cytoplasm. But there are other reasons. These cells have so-called intracellular canaliculi, which are visible only in the electron microscope. These are deep invaginations of the plasma membrane, and that's why the cell boundary becomes highly irregular. We cannot appreciate it in the light microscope, but it is visible in the electron microscope. So increases the cell surface area, because in an increased cell surface area, more pumps can be embedded into the membrane, so production is quicker. I mean, hydrochloric acid production is quicker. Yes?
Is it possible that because of the high concentration of protons, it's more eosinophilic?
It could be, but don't forget the chloride ions.
So
So they keep all, yeah. So it cannot be the reason, therefore. There must be something else. And this is the mitochondria. And also this irregularity and this very big total surface area, because the membrane area becomes big, and the membrane, we have lots of intramembranous proteins, and altogether these increase the intensity of the eosinophilia of the cell. And the third thing, tubulo-alveolar vesicles, tubular vesicles are in the cytoplasm, so elongated vesicles which contain extramembrane pieces. And those extramembrane pieces, again, full of proteins, pumps. And because they are stored in large amount inside the vesicles in the cytoplasm, it also contributes to the eosinophilia. And we have discussed one cell in the previous semester, which also stores prefabricated membrane pieces in vesicles. Which cell was that? Those extra pieces are called plugs, but what is the name of the cell?
Transitional epithelium.
In the transitional epithelium, but which cell? Umbrella cells, correct. Umbrella cells. So we try to interrelate things because we try to know enough. And if we can interrelate different things, it is much easier to remember for. Okay. So these cells responsible for hydrochloric acid production, but they produce something else as well. Intrinsic factor, which is a glycoprotein. Again, protein-like structure. Which is needed for absorption of vitamin B12 in the ileum. Without this, the vitamin B12 cannot be absorbed, and that may cause problems in a patient, may cause anemia, because red blood cell production needs vitamin B12. So if somebody has an atrophy in the mucosa of the stomach, so these cells tend to disappear, will not produce enough intrinsic factor, those patients need to have regular B12 injections to supply the patient with enough B12 to prevent the patient from anemia. Okay, so these are the parietal cells which dominate in the middle part of the gland. Now, if you go down a little bit, well, actually not a little bit, but to the bottom of these glands, here, most cells has basophilic cytoplasm, like here. Some parietal cells are still here, but basophil cells dominate. Now, these cells with basophilic cytoplasm are the chief cells, which produce pepsinogen, which is an inactive precursor of the active enzyme called pepsin. And pepsin is responsible for cleaving proteins. And it is produced in an inactive form, like pepsinogen, because if it was produced in its active form, then the pepsin would digest the tissue of the lamina propria, tissue of the stomach itself. Self-digestion would occur. And actually, hydrochloric acid is going to the lumen of the stomach, just like the pepsinogen. And in the lumen, pepsinogen, due to the low pH value, it will be converted only there into the active form, into pepsin. And also hydrochloric acid is appearing here by combination of the chloride and hydrogen ions. So here in the lumen of the stomach we have a very drastic environment which could harm the tissues. So the tissue of the stomach needs defense against this low pH environment and against the enzymes. And this defense is given by this high mucus production. So remember, all surface cells produce mucin, in addition to lots of mucous neck cells also produce mucin. And this mucin is alkaline, so it may act as a preventive mechanism against the acidic environment. Okay, so we have talked about 3 types of cells in the gland: mucous neck cells, parietal cells, and chief cells. There are 2 more, but we cannot distinguish with hematoxeniosin staining. These are the endocrine cells, which produce hormones like gastrin and many more. You may call them as enteroendocrine cells as well. And the other cell type is the undifferentiated cell. Undifferentiated cells are mostly in the neck region. They are undifferentiated multipotent cells which can give rise to multiple cell types which are present in the gland. Now this is the cellular composition of the fundic glands or glands of the body. Okay? And I mentioned that this is different in the cardia and pyloric region, and the difference is that in the cardia and in the pyloric regions, parietal cells and chief cells are mostly missing. So the other cells are there, but parietal cells and chief cells are very rare or absolutely absent in the other two regions. So that's why we can say by just looking at this section that this must be from the fundic region or from the body region. Yes.
Can you show the differences between them?
In another section, we will see the pyloric region. We don't have section from the cardiac region.
No, but to see the chief and parietal cells.
Okay. Parietal cells are eosinophilic, and chief cells are basophilic, like these. These are parietal cells 1, 2, 3. Others here are chief cells.
But pepsinogen is also a protein, no?
Hmm?
Pepsinogen is also a protein, no?
Yes, and protein production needs lots of RNA, and that's why the basophilic cytoplasm.
So it has more RNA than the amount of actual proteins inside.
Mm-hmm. That's why the balance is shifted. Okay. So muscularis mucosae, fine, and underneath we have submucosa, connective tissue. Again, we have submucosa plexus. We have lymphatic vessels like here. Of course, blood vessels as well. Well, muscularis mucosae, maybe a little pieces here. Submucosa plexus here. Submucosa plexus. Then we have external muscle layer, which is divided into 3 sublayers. It is usually described as an inner oblique layer, a middle circular layer, and an outer longitudinal sublayer. Well, it assumes that we have a well-defined axis, what we don't really have in case of the stomach. But it is quite visible that the orientation of cells here or here or here are different, so they are differently oriented. And within this external muscle layer, we have again the enteric plexus. It is more developed here already. Here. Look identical to that what we could see in the esophagus. Yes. Well, it looks the same, but more underdeveloped. Here, maybe this is another piece of this submucosa plexus, just nerve, peripheral nerve here. Okay. And the outermost tissue layer, because this is intraperitoneal organ covered entirely by peritoneum, unlike the esophagus. Here we don't have any adventitia. Instead, we have subserosa and serosa. So subserosa is a loose connective tissue layer where blood vessels and nerves reach the organ. And of course we have connective tissue fibers and connective tissue cells as well. And the very peripheral part, we should be able to see mesothelial cells. A simple squamous epithelium consisting of mesothelial cells. The problem is I have difficulty to show them. Maybe I can show one here, because most of them were lost during sectioning. Here another one. So mesothelial cells have squamous heterochromatic nucleus like this. So that's a special simple squamous epithelium, what we call mesothelium, and they consist of mesothelial cells. So this mesothelial layer is the serosa, and the underlying loose connective tissue is the subserosa. This is the case whenever we have an organ which is covered entirely by a serous membrane. So this is the organization of the wall here. We have another stomach section with hematoxaniosin staining. It is a little bit darker, so blends a bit differently appearing, but their sense is the same. So you can see these parietal cells, for example, very well. And you can distinguish them very well from these chief cells, which have basophilic cytoplasm. And mucous neck cells, well, here are the mucous neck cells. Look at the cytoplasm, the color, try to memorize. And if you go to the surface, well, the surface cells have a little bit different shade, again, because of the different mucin they produce. Okay, so essentially the same, maybe one extra here. This is a lymphocyte accumulation, also another one here. These are called lymphatic follicles or nodules. Inside we have a pale stained center called germinal center. If we have such a germinal center, it is a secondary lymphatic nodule. If we don't have it, is a primary lymphatic nodule. We will learn about them more later. This lymphocyte accumulation, and actually the lymphocytes and other immune cells appearing in the lymphoreticular connective tissue of the lamina propria, also belong to the so-called diffuse lymphatic system. So diffuse because they are not localized in dedicated lymphatic organs like lymph node or tonsils or spleen or thymus or bone marrow. And this diffuse system is associated with the mucosa. Therefore, we can call it mucosa-associated lymphatic tissue. Abbreviation is MALT, M-A-L-T, mucosa-associated lymphatic tissue. But we can define subdivisions within the MALT depending on the organ system where the mucosa belongs to. So this belongs to the alimentary canal. So we can call this subdivision as gut-associated lymphatic tissue. Gut is intestine, so in broader sense, alimentary canal. So this division of the MALT is called GALT, gut-associated lymphatic tissue. I previously showed lymphatic nodule in the wall of the trachea as well. So that's in the mucosa of the respiratory tract. It is called BALT, bronchus-associated lymphatic tissue, BALT. That's another subdivision of the MALT. So MALT has two major subdivisions, BALT and GALT. Okay.
That is the only tissues that we have mucosa in the respiratory tract.
No, we have in the urinary system as well, so that's a third subdivision, if you like. Now we have a stomach section with PES plus hematoxylin. I will open this 46 underline 2. So no eosin is here, that's an important fact to emphasize. So PES, periodic acid shift, is a histochemical reaction, what we learned. It can mark carbohydrate-rich territories in purple color. In addition, we have hematoxylin dye. So essentially the same. So I just will concentrate on the structures which gain PES positivity. So which are marked by the PES reaction because of the high carbohydrate content. That's why they will become purple, because otherwise I could repeat everything else. So what are the PES positive structures here? So content of the stomach, which remain, is PES positive because it contains lots of mucin, right? Now, these surface mucous cells, the simple columnar mucus producing epithelium, all cells of this epithelium also PES positive because they produce mucin. Inside them we have lots of mucin. Of course, we used alcoholic fixative here, not formalin, to preserve the mucin and the carbohydrates in general, to be able to detect them. So these surface cells also PS positive. Now, here are the glands, and these are the mucous neck cells here, and they are a little bit paler than the surface cells. These also produce mucin, these mucous neck cells, but different kind of mucin, that's why the different shade. If you magnify the lamina propria, here inside the lamina propria in the lymphoreticular connective tissue, you can see purplish line-like structures. Those are the reticular fibers, which are PS positive because consist of type 3 collagen molecules mostly. And type 3 collagen is the type of collagen which is associated with the most carbohydrates. Enough carbohydrates which can show PS positivity. Other collagens are also associated with some carbohydrates, but not so much. So that's why only the type 3 collagen is PS positive. So only the reticular fibers are PS positive. Of course, if we can see the basement membrane, if we could see, maybe here, some sign, can you see this line, maybe, underneath the surface epithelium. It is also PS positive because contains a reticular fiber layer, which is from reticular fibers. Okay, now, if you look at the parietal cells, they are not red. No red cell at all, because no eosin is here. So this is a parietal cell. Their cytoplasm is pale, or this one. Okay, but chief cells are basophilic because hematoxylin is present. Okay. Okay, we reached the muscularis mucosae, smooth muscle, PS positive slightly. Why? Why?
Because the glycogen in the smooth muscle?
Because in the smooth muscle we have lots of glycogen which is needed for production of energy via glycolysis. So generally muscle cytoplasm is PS positive slightly. That's why the wall of vessels is also PS positive, because in the tunica media we have smooth muscle cells. Right? Now, muscularis externa, again smooth muscle, slightly PS positive. And finally, if we have a territory where mesothelial cells are preserved, which is rare, as I said, because we tend to lose them. So maybe it's difficult to have an example here at all. Well, maybe one. So actually what I would like to say that underneath the mesothelium there is a basement membrane which separates it from the connective tissue layer of the subserosa. And that's why this is a PS positive layer, because that's the basement membrane inside which we have reticular fibers. So basement membrane everywhere is PS positive, and we have a basement membrane here as well, which separates the mesothelial layer from the connective tissue of the subserosa. This is another mesothelial cell.
So the basement membrane is between the serosa and the subserosa?
Basically, yes. Well, that's again a problem with nomenclature, you know, because some books consider serosa exclusively as the mesothelial cell layer, as I said. But some other books consider serosa as the mesothelial layer plus the immediate territory of the connective tissue layer together, and then the rest of the connective tissue is termed as subserosa. So precisely between the mesothelium and the connective tissue, there must be a basement membrane. But broadly speaking, yes. Okay, so as you can see, We are far from being complete, so please remind me on Friday that we have still the pylorus duodenum transition plus jejunum, yes? They produce different kinds of mucin, so the chemical composition is not the same, and that's why the staining effect is slightly different.